is Morten Scheibye-Knudsen, who is a researcher in aging biology in the University of Copenhagen. Morten works across the spectrum of aging research and is also very well known as the co-organizer of a conference in Copenhagen, Aging-Related Drug Discovery, or ARDD, which is possibly the most famous and well-attended conference in the aging field, and one I think you should all have a look at registering for at some point. So please welcome Morten to the stage. Thank you.
Very impressed about the whole organization here. It's really a beautiful place, fantastic venue, so thank you so much. And so I'm going to talk a little bit about brain aging, which is actually not a topic that we work too detailed on, but I'm going to give it from our perspective. Okay? So my lab really focuses on understanding the basic biology of aging, so and particularly what is driving the phenotypes that we see with aging. So in essence, you could say, why do we develop graying of hair? Why do we develop facial wrinkles? Why do we develop redistribution of subcutaneous fat? And all these phenotypes that are associated with aging. So these are, of course, the phenotypes just mentioned are just phenotypes that you see on the surface, and there's, of course, a lot of stuff also going on the inside. But what's quite interesting is that our face is actually a very strong biomarker of aging, I would say. Maybe it even reflects biological aging, although Matt may disagree with this. But it turns out that you can develop aging clocks based solely on facial photographs. And so in this has been done before, but in this study we did facial photographs upon ER admission on a relatively large number of patients in the Copenhagen area. And then we have follow-up data so we can actually develop algorithms to predict survival also. And it turns out that our face is really strong. So if you look at the bottom left graph, this is the age predictor. It's relatively good at predicting someone's age, but it's also extremely good at predicting survival. So if you look at the bottom right graph, you can see that overall survival is strongly dependent on how your face actually looks. And it's actually a stronger predictor than any blood sample that's taken upon ER admission. It's a stronger survival predictor than your blood pressure, pulse, saturation, etc. So this is something that I think could be very easily implemented in a trial setting in the ER, but potentially also as a general physicians at your family physician.
That's hard. That's impossible.
Our surface phenotype, our face is of course only one thing that changes with age. There's a lot of other things that change with age. In this particular study, we downloaded PubMed, which is this very large database of biomedical literature. And then we tried to ask the question, what is actually the phenotypes that are associated with aging? And the phenotypes are really very varied. So the phenotypes that we dug out from PubMed, we then correlated this in cohort studies. And we can see the graying of hair is the most prevalent feature that we see in aging. 99% of everybody will develop graying of hair. But there are other features that are, I would say, much more important. So muscle weakness is a very strong phenotype that you see with age. Cancer occurs in about 40% of everybody, and between 10 and 20% of everybody will develop some type of dementia. So aging is very varied across across humans. In fact, the older we get, the more different we look phenotypically. And so another set of diseases that we've been interested in is premature aging diseases. These are genetic diseases where you have a single mutation in a gene that leads to accelerated aging of individuals. And so there's a long list of these diseases that describe particular features of aging. So some of them have accelerated vessel aging. This is particularly Hodgkin-Gilford's progeria. Some of them are more cancer syndromes, such as Bloom syndrome. And then there are others that are that have features of brain aging. This is diseases such as Cockayne syndrome, ataxic telangiectasia, or some types of xeroderma pigmentosus. These are extremely rare diseases, and there's a large group of these diseases with different phenotypes that show overlap with human aging. So how do we investigate which of these phenotypes are important? So one thing that we've done before is to actually look at how do different phenotypes associate with each other. And because we can quantify the phenotypes based on the prevalence of that phenotype in a different disease, we can see how different diseases overlap in phenotypes and how they associate with each other. So this is a heat map where we can see how the different diseases associate with each other. So the columns are diseases and the rows are phenotypes. And you can see that there's a cluster of phenotypes that involve short stature, microcephaly, developmental delay. These phenotypes co-segregate in these genetic diseases, suggesting that they probably share some sort of origin in their development. That's our hypothesis. In fact, we've done some old work on this, showing that some of the premature aging diseases show similarities with primary mitochondrial disorders. And these are particularly the diseases where you see accelerated brain aging, suggesting that the mitochondria may be particularly important for the brain. These diseases, the premature aging diseases, are almost exclusively caused by genes involved in DNA maintenance. So if you accumulate damage in your DNA, it appears that you age faster. But all damage is obviously not equal. Some damage appears to lead to mitochondrial dysfunction and a phenotype that can be considered similar to primary mitochondrial disorders. And some of the work is shown here. So we found that when the DNA damage response is activated in the cell, that can lead to activation of an enzyme called PARP1. And that's an enzyme that polymerizes NAD, so it removes NAD basically. And that leads to loss of NAD and metabolic changes and contributes likely to the neurodegeneration. And so we can inhibit PARP1 using PARP1 inhibitors. These are also used in the clinic for cancers. But you can also go in and actually take a small molecule, a supplement. We heard about NMN earlier from Brian, but you can also use something called NR, nicotinamide riboside, which is a molecule. It's a small molecule, it's a supplement that gets converted into NAD. And so this has received quite a lot of interest in the field since this was explored. And recently we actually ran a trial on individuals where we expect there's more DNA damage. And so this is actually COPD patients. So when you smoke, you induce damage in your lungs. And we hypothesized that that damage could lead to loss of NAD. And so we ran a placebo-controlled, double-blinded, randomized clinical trial to explore if NR could influence COPD. And we chose our outcome measure as inflammation because these were stable COPD patients. It's a short trial, and it's been shown before that NR may impact inflammation. And in fact, that's what we found. So we found the COPD patients had lower levels of NAD, consistent with potential and increased DNA damage response. And the amount of NAD correlated quite strongly with the amount of lung function. And when we gave them NR, we saw that NR significantly reduced inflammation in the lung. So that was quite exciting. And so damage is not equal. There are different types of DNA damage that can lead to different outcomes. So you can consider three major outcomes of DNA damage. If you damage a cell, either the damage will be so strong that the cell will undergo apoptosis. It can transform and become senescence and a pro-inflammatory cell, which may be involved in this COPD pathway. Or if the damage is repaired, it could lead to mutations and tumorigenesis. And so there's been a lot of work considering if mutations may contribute to aging. And so the apoptosis senescence is sort of a damage signaling. You do not get apoptosis or senescence unless damage signaling is present. And that, I think, is probably involved more in degeneration. So neurodegeneration or other types of like muscle loss with age is probably due to signaling that leads to degeneration. Conversely, mutations is really what's driving tumorigenesis. So we asked, and this is unpublished work, it's actually the first time I'm presenting this data, I hope. This is not working, so, oh, there we go. So, we asked, one thing that you noticed, one thing that we asked was whether or not mutations could accumulate with age and how they, and when they accumulate. And the way we did this was to look at single-cell RNA sequencing data. So if you have a single cell, you do RNA sequencing in single cells, there will be an RNA copy of the DNA strand, and that copy will actually reflect the underlying mutations in that cell. And because there are published data sets from individuals from multiple tissues, we can actually dissect where do these mutations come from. Are they present at conception, basically, or do they actually accumulate during development, or are they age-associated? So this was the question that we did here, and this is in work in collaboration with Eric Verden at Buck. And so our first surprising finding was that looking at mice and human data, we found that the majority of mutations that accumulate during lifespan actually accumulate during early development. Okay? And we can quantify that here in these pie charts, particularly for the mice, where we can see that only 10% of the total mutational load that a mouse will accumulate during its lifespan is actually age-associated. So that suggests that mutations are probably not what's driving functional decline with age, but it could be what's driving, for example, cancer development. This fits very well with premature aging diseases. We're also using our sort of phenotypical knowledge to dissect new premature aging diseases. So in this particular project, we used our aging phenomes, our collections of phenotypes with age, and then we asked, can we find new premature aging diseases? And we went into the OMIM database, which is a database of genetic diseases, and then we just basically looked at diseases that showed phenotypical overlap with human aging. And our surprise was that we found a top hit called Woodhouse-Sakata syndrome, which is a previously undescribed disease, and that showed strong overlap with human aging. So what is Woodhouse-Sakata syndrome? So it's a rare autosomal recessive disorder. It's caused by mutations in a gene called DCAF17, and the patients, when you just look at them, they actually look kind of prematurely aged. And so we did this phenotype clustering, and we saw that Woodhouse-Sakata syndrome associates strongly with human age. We also used our neural networks to determine the perceived age of the individuals, and they also were predicted to be older. And we were very fortunate that the first, or fortunate, can you say, but the first family of these patients in Northern Europe are in Copenhagen, and we got blood samples for them and used another biomarker of aging and found that they were also predicted to be older with this biomarker. We created a mouse model. The major phenotype of that mouse model was that the mice were sterile, but they also showed increased inflammation and frailty with age. So it's probably, we think, a bona fide accelerated aging disorder. So DCAF17, what does it do? So the rest of the premature aging diseases are repair disorders. So these are cells here where we have expressed DCAF17. I think there should be a video. It doesn't work. Okay, trust me. If you shoot the cells with a laser, DCAF17 gets recruited to the laser site. We can also do mass spec and look at the proteins that are associated with DCAF17. So we pull down DCAF17, and then we find members of a repair complex called DDP1, called 4-RBX complex, which is involved in nucleotide metabolism. We can knock out or create mutations in DCAF17, and we find specific mutations that are important for this interaction. We can also then try to further understand the mechanism on why, what is DCAF17 actually doing in this complex. And so for this, we did an assay where you fuse DCAF17 with a ubiquitin ligase, and then you can identify which proteins are recruited around this enzyme. And we find this TRAP3 protein that are involved in this. And we can see that it actually gets, that the DCAF17 seems to stabilize TRAP3. And what is TRAP3 then? So TRAP3 has been involved in maintaining something called R loops. So R loops are RNA-DNA hybrids that accumulate in our genome. And so we want to remove them. So TRAP3 has been shown to be involved in facilitating the removal of these R loops. And so it would make sense that if DCAF17 stabilizes TRAP3, you get easier R loop removal, and then less of this sort of genotoxic stress and less premature aging. And in fact, we can see that there are more R loops. We can also see that there's more single-stranded DNA using various markers, suggesting that, that DCAF17 does indeed impact R loop metabolism. And so DNA metabolism, changes in DNA metabolism can lead to a lot of different phenotypes. These are kind of listed here. We think that the two main ones that are impaired in R loop metabolism is probably this replication issue. So if you have accumulation of these R loops, you have an issue with replicating your DNA, and that's why the patients have issues with, for example, hair loss and hypogonadism, so loss of fertility, particularly for males. And potentially also this DNA damage signaling that then drives the neurodegeneration that you also see in these patients. So DNA repair seems to be important in aging. So what if we could stimulate DNA repair? What if we could identify molecules that can actually activate DNA repair? Then maybe we would be able to treat some of these age-associated diseases. And so we're doing that a little bit. This is actually a project that's been going on for a long time, that was started in collaboration with Silico Medicine, a company that many of you probably knows. And the idea was that that we could take advantage of this adaptive response called hormesis. We're going to hear about that from Suresh, I hope later in this conference. And the idea is that if you stress cells and the cells do not die, they will adapt and become stronger. So we looked at that for molecules that could actually hijack this response and use it for our advantage. And so we screened a very large amount of molecules, 15,000 molecules, to identify molecules that would mimic the DNA damage response. And so our first test was looking at the top hits to see if they could protect against ionizing radiation-induced cell death. So each dot here is a petri dish that have been treated with different compound, and then we hit them with a giant X-ray hammer, and then we see which of the cells are actually surviving. And we find that some of the petri dishes, the cells actually become completely immune to ionizing radiation, and some of them are sort of adapting to some extent. The ones that are completely immune are typically the ones that have a very, very severe initial effect of the compound. So you see strong cell death. So those one we actually did not want to pursue. But then we wanted to pursue drugs that didn't by themselves induce DNA damage. So they would elicit the hormetic response without doing DNA damage. And we did that in various ways. So this is a COMET assay where we looked at DNA breaks. So it's basically each dot here is a nuclei. And we apply an electrophoretic field across it, and then if there are breaks in the DNA, the DNA will be pulled out of the nuclei, and you get this sort of comet-like shape. And we find some of the top hits did induce DNA damage, and some did not. Another way to look at it is using these markers, 53BP1 or gamma H2AX, and we did find some drugs that led to a strong DNA damage response, but we actually also found some that did not. In particular, we found this drug F that did not. And so we tested many of these drugs in various assays. So this is an assay for repetitive senescence, which is basically if you have primary cells grown in culture over a long period of time, at some point they will stop dividing. This is called the Hayflick limit. And there are various markers associated with senescence. And we see that one of the strong markers of senescence is lamin B1. If we treat with this compound, we increase lamin B1 expression. We also reduce beta-galactosidase, and we increase the repetitive lifespan of these cells. And in fact, if we look at DNA damage markers, which are thought to be actually quite stable markers during repetitive senescence, we actually see a reduction in these DNA damage markers. We can treat fruit flies with the compounds. This was done in collaboration with Tract Biotechnologies, which is a spin-out from my group where this company can do motor tracking. We can see that the flies that are, when you're a young fruit fly, you move a lot. If you're an old fruit fly, you move less. But if you're an old fruit fly on drug F, you actually still move quite a lot. And you see this quantified in the middle, and you also see a small extension of lifespan in these flies. So we tried to identify molecules that were similar in structure that could elicit the same response. So we screened about 30, 40 different compounds. And then we tested how they would impact senescence. And we found a number of molecules that actually reduce cellular senescence and actually allow senescent cells to become not senescent anymore. So they actually reverses senescence. So this is quite interesting. And our lead compound, we then went into a mouse model of Alzheimer's disease, because we know in Alzheimer's disease there's more DNA damage in the brain. And so we treated these mice with this compound. And this was kind of a side finding in the project was that so Alzheimer's disease mice are actually leaner than normal mice. They grow a little bit slower, but when you treat them with this lead compound, they regain the same body weight as wild-type mice. We also rescued the motor function deficits that you see in the Alzheimer's disease mice. And when we look for memory, we rescue the memory deficits in the Alzheimer's disease mice and also reduce the amount of DNA damage, that's the bottom here, in the mice. So we're actually spinning this out as a company. So it's still in the process. So and of course it needs a lot more development. But I think the take-home message is that DNA damage signaling is, I think, very important for aging, and particular in the context of degenerative diseases. And we can actually target either the consequence of DNA damage using molecules such as NR, or potentially DNA damage by itself. And so there are many, many trials underway, and I think this is a very exciting field. We've heard a lot about how the field is progressing into clinical trials, and I'm very excited about that. And I'm just going to finish here and thank the people. This is actually an old picture, but thank the people in my lab. Thank you for listening, and if there are any questions, I will be happy to take any questions.
Thank you. Thanks, Martin. Thanks very much. So I believe we have a couple of questions here we might take a quick look at. Thank you for an explanation of some of the science that's taking place. So I don't know if you want to pick one of these, but
anything.
There's some good questions there. So what practical steps can clinicians take to slow brain aging in patients with multiple comorbidities?
So this is some of the stuff that we already know. I mean, so I think some of the strongest data on driving better brain health is actually movement. So there's data showing that even if you have beginning cognitive decline, if you start exercising, you can actually reverse a little bit of that decline and maintain brain health later. And There's data from Dutch group showing that exercise also increases NAD levels. This is completely circumstantial data, but maybe that's involved in it. There's also trials now underway using NR, NMN in both Alzheimer's disease and Parkinson's disease, and we'll see how that works. I think that there's a lot of negative data with NR, but I think there's also some data showing that NR reduces inflammation. This is also what we've seen in our placebo-controlled trial. I think the data was relatively convincing there. So I kind of believe this inflammatory effect. And we know that inflammation is also probably driving some of the phenotypes that we see in, for example, Alzheimer's disease. So,
okay. Similarly, what are the strongest biological drivers linking aging to accelerated brain disease? And are they actionable via lifestyle and behavioral interventions? Again?
Yeah, I mean, so I picked the DNA damage. This is my completely unbiased view. But obviously, there's a lot of other stuff. We've heard about the hallmarks. I think proteostasis is very important for brain aging. We know that protein aggregates accumulate in age-associated brain diseases. So, and it's difficult to kind of go into the root of that to affect that, but you can, I mean, there are treatments now, right, where you can remove some of the amyloid beta load, and that seems to reduce some of the phenotypes that you see with Alzheimer's disease. But the effect is really, I think, relatively small. So I'm not sure if this is like a root cause. I think this is maybe a bystander that comes a little bit later, and hopefully we can affect some of the root causes. And my unbiased opinion is that DNA damage could be some of this.
Amazing. Morten, thank you very much. Give him a round of applause. Thank you.